This video is for A-level biology students to help them practice for their exam. So we'll be doing practice of paper 1. So let's start our first question. Figure 1 shows a cell from the lining of ileum specialized for absorption of products of digestion. SGLT1 is a carrier protein found in the cell surface membrane of this cell. It transports glucose and sodium ions into the cell. So that was just the background of the question. So the question is, the action of the carrier protein X in figure 1 is linked to a membrane-bound ATP hydrolase. Explain the function of this ATP hydrolase. So basically what we see here is we have an ileum cell, and here we can see a carrier protein. And as mentioned in the question, this protein needs the ATP hydrolase to function. So ATP hydrolase will basically hydrolyze the ATP molecule into ADP and releasing energy. So your answer should cover both the point. The first point you need to cover is that ATP to ADP plus inorganic phosphate releases energy. And number 2, you would say that energy allows ions to be moved against a concentration gradient, or you can say that energy allows active transport of the ions. So for the carrier protein, obviously we need energy, and ATP hydrolase enzyme helps us by hydrolyzing ATP to ADP that releases energy. So it was a simple question. So let's move on to the next part of this question. The movement of sodium ion out of the cell allows the absorption of glucose into the cell lining of the ileum. Explain how. Before you answer this question, you need to understand the concept. So let's say we have this ileum cell, and this is the lumen site of the intestine, and on this side we have the blood vessel. Digested glucose will be here. So the first step is that glucose should be absorbed into the cell, and the second step would be that the glucose must be transported from the ileum cell into the blood. Now the problem is glucose is a big molecule and it can't pass through the membrane, so it needs some carrier proteins. So let's see how it works. So here we have the ileum cell. So the first step is to get rid of the sodium inside the cell, and for that purpose the sodium potassium pump is used. So sodium is removed from the cell, and that decreases the sodium concentration inside the cell. So now we have more sodium outside the cell compared to the inside, and that establishes a sodium concentration gradient from outside to inside. So if you look here, we have here a sodium glucose cotransporter. So what this does is transports sodium into the cell, and every time sodium moves inside the cell, it brings glucose molecule with it, and that's why it's called sodium glucose cotransporter. In the next step, the glucose must be removed from the cell into the blood, and for that purpose protein channel is used, which gets the glucose out of the cell into the blood. So this is how the glucose is absorbed into the cell and then released out of the cell into the bloodstream. And this all was possible because of the sodium ion, because sodium co-transported glucose into the cell. Again, this sodium must be removed from the cell so that there is a higher concentration of the sodium outside the cell compared to the inside, and that will maintain a flow of sodium from outside to inside, and sodium would therefore be able to move glucose with it. Okay, so now how you will answer this question, because this is for 2 marks, so you need to cover these 2 points. So number 1, you will say that it maintains or generates a concentration or diffusion gradient for sodium ion from ileum into cell. And number 2, sodium ion move in by facilitated diffusion brings glucose with it, or you can say sodium ion moving in by co-transport brings glucose with it. Next question. Describe and explain 2 features you would expect to find in a cell specialized for absorption. And that's for 2 marks. Of course, if the cell is involved in absorption, it must have a large surface area so it can absorb. And then secondly, it must have those carrier protein channels so it can absorb easily. So your answer should include these 2 points. So the cell should have folded membranes or microvilli to provide the large surface area for absorption. And number 2, you would say large number of co-transport carrier or channel proteins for a fast rate of absorption. Or you can say the same thing in a different way. Or you can say a large number of mitochondria, so make more ATP by respiration, and that will provide energy. Or you can say in a different way. And finally, you can say membrane-bound digestive enzyme, so maintains concentration gradient for fast absorption. So you can mention any 2 of these points. Let's move on to the next question now. Figure 2 is a diagram of one SGLT1 carrier protein, and that's a figure 2. So as you can see, it's a quite big protein. It's got amino group on this side, and it's got a carboxylic group on this side, while this region is a hydrophilic region, and this is the hydrophobic region. Okay, so now what the question is. So the question is, draw phospholipids on figure 2 to show how the carrier protein SGLT1 would fit into the cell surface membrane, and do not draw more than 8 phospholipids. And that's for 2 marks, and it's an easy question. So all you have to do is just show in the figure how this will fit into the cell membrane structure. So this is how it should look like. So here is the SGLT1 protein. And as you can see, it's embedded in the phospholipid bilayer. So we're showing 4 on this side and 4 on the other side, because it mentioned the question not draw more than 8. And if you remember, this head is hydrophilic and these tails are hydrophobic. And this part of the protein is hydrophobic, so that should match with the hydrophobic part of the membrane. Okay, so now let's move on to the next question. Figure 2 shows the SGLT1 polypeptide with NH2 at one end and COOH at the other end. Describe how amino acids join to form a polypeptide. So there is always NH2 at one end and COOH at the other end. You may use a diagram in your answer, and that is for 2 marks. To answer this question, you must understand how the amino acids join together to make a polypeptide. If you look at an amino acid, it's got 2 ends. On one side we have the amino group, while on the other side we have the carboxylic group. So that is one complete amino acid. Then we have another amino acid. These amino acids can join together by making a peptide bond, but that is a condensation reaction. So that means to make that peptide bond, a water molecule will be released. So this is where the water molecule comes from. So the OH comes from this side and hydrogen comes from the amino group side. So the water molecule will be removed and a bond will be formed between the carbon and the nitrogen, and that will be our peptide bond. So in the same way, all the amino acids will join together to make a long protein molecule. So if you look carefully, you can see that there's an amino group on one side and carboxylic group on the other side. Because this question was for 2 marks, so your answer should include these 2 points. So you will say, number 1, one amine or NH2 group joins to a carboxylic group to form a peptide bond. Number 2, so in chain there's a free amine group at one end and a free carboxylic group at the other end. Or you can say that each amino acid is oriented in the same direction in the chain. Okay, so that was question 1. Now let's move on to question number 2. So the question 2 is about a study. A scientist placed a tube into the gut of a healthy 20-year-old man. The end of the tube passed through the stomach but didn't reach as far as ileum. So that was still in the stomach. It didn't go down to the small intestine. The scientist fed the man a meal containing triglyceride through the tube. So that person was given triglycerides, which is a type of fats. The scientist also used the tube to remove samples from the man's gut at intervals after the meal. So he was fed and then he removed the samples after some regular intervals of time, so he can see how much triglycerides have digested. The scientists measured the type of lipid found in the sample. Some of the results are shown in table 1. Okay, so here we have the table with the results. So we had 2 different samples. So the first is the sample A. The first column is time of collection after meal, and that's in minutes. So the first sample was taken after 45 minutes. The second column says the concentration of the fatty acid. For example, in the first case, it was 2.7, and the third column says concentration of triglycerides, and in this case, it was 0.6. Sample B was taken at 75 minutes, and in this case, the concentration of fatty acids was 3.3. So that means the concentration of fatty acids has increased, while the concentration of the triglyceride is 0. So that means there are no more triglycerides. And the reason is pretty simple, because the triglycerides have been digested and they have converted into fatty acids. So that's why we see more fatty acids at 75 minutes. Okay, so now what the question is. So the question is, use your knowledge of lipid digestion to explain the differences in the results for sample A and sample B shown in table 1. You should assume that no absorption had occurred. And just for information, so this is how the triglycerides look like. So when they are hydrolyzed using the enzyme lipase, they are digested into glycerol and fatty acids. So this part here is the glycerol, while the rest of this is the fatty acids. So the 1 glycerol and 3 fatty acids molecule. And we can see this clearly from the table that the concentration of triglyceride has gone down to 0 because they have all been digested, and the concentration of the fatty acids has increased. So your answer should include these 3 points. Triglycerides decrease because of the action of lipase, or you can say fatty acids increase because of the action of the lipase. Second point, you will say triglycerides decrease because of the hydrolysis of triglycerides. And as I mentioned before, there is a hydrolysis reaction because a water molecule is required to complete this reaction. Triglycerides decrease because of the hydrolysis of triglycerides, or you can say fatty acids increase because of the hydrolysis of triglycerides. The third point you will say is the triglycerides decrease because of digestion of ester bond. So ester bond is basically the bond between the glycerol and fatty acids. So I've just told you that is the glycerol part and this is the fatty acid. So this bond between the glycerol and fatty acid is the ester bond. So triglycerides decrease because of the digestion of ester bonds between fatty acids and glycerol, or you can say fatty acids increase because of digestion of ester bonds between fatty acids and glycerol. Next part of this question is, after collecting the sample, the scientists immediately heated them to 70 degrees for 10 minutes. Explain why. And that question is for 2 marks, so you should cover 2 points. So the reason scientists immediately heated them at 70 C is to inactivate the lipase, otherwise the lipase will continue its action. So you can say to denature enzyme lipase. And number 2, so no further digestion, hydrolysis or catalysis occurred. The next part of this question is, describe the role of micelles in the absorption of fats into the cells lining the ileum. Before you answer this question, you should be clear what the micelles are. So basically, fatty acids and monosaccharides are emulsified by bile salts to form micelles. Because fatty acids and monosaccharides, they are not soluble in water, so it's hard to absorb them. So it's the bile salts which emulsify them and make them water soluble, and then they are easily absorbed through the epithelial cells. So your answer should include any 3 of these points. Micelles include bile salts and fatty acids. And number 2, you can say make the fatty acids more soluble in water. Number 3, bring or release or carry fatty acids to cell lining of the ileum. Number 4, maintain higher concentration of fatty acids to cell or lining of the ileum. Number 5, you can say fatty acids absorbed by diffusion. Okay, so let's move on to the question number 3. Figure 3 shows pressure and blood flow during the cardiac cycle in a dog. So here we see 3 graphs. So the top one is pressure in the left ventricle. Then the bottom one is pressure in the aorta near the heart. And the bottom one is rate of blood flow in aorta near to heart. Okay, so the question is, at P on figure 3, P mean at this point, the pressure in the left ventricle is increasing. Yes, we can see that the pressure is increasing. At this time, the rate of blood flow has not yet started to increase. So if you look at the rate of flow, which is at the bottom one, so at this point, if you keep coming down here, this point, so the rate of blood flow is still not increasing. Yes, we can see that. Use the evidence from figure 3 to explain why. So the question is that the pressure in the ventricle is increasing, but the rate of flow in the aorta is not increasing yet. So what is the reason for that? But first, have a look at the diagram to help you understand. So basically, we are talking about the left ventricle, which is here. So when the left ventricle contracts, the blood is forced into the aorta, which is here. And keep in mind that between the left ventricle and the aorta, there is a semilunar valve. So when the pressure in the left ventricle increases, that forces the semilunar valve to open. So obviously the valve will open only when enough pressure has been generated in the left ventricle that will open the semilunar valve. So your answer should include these 2 points. So number 1, aortic or semilunar valve is closed, and number 2, because pressure in aorta higher than in ventricle. So the next part of this question is, at Q on figure 3, there is a small increase in the pressure and in the rate of blood flow in the aorta. Explain how this happens and its importance. So that's questions for 2 marks. So we need to look at the figure again. Okay, so point Q is here and here. And we can clearly see there is a small increase in the blood flow here and here. And now the question is, why is that? So basically what's happening here is because the aorta is elastic, so it's just the elastic recoil that generates this small blood flow. So aorta is a major vessel and it's the elastic recoil after every heartbeat which makes a small flow. So your answer should include number 1, elastic recoil of the aorta wall or tissue, and number 2, smooths the blood flow, or you can say maintains a rate of blood flow, or you can say maintains blood pressure. The next part of this question is, a student correctly plotted the right ventricle pressure on the same grid as the left ventricle pressure in figure 3. Describe one way in which the student's curve should be similar to and one way it would be different from the curve shown in figure 3. So when you're plotting the pressure of the right ventricle and the left ventricle, the right and left ventricle contract at the same time, so they both should start at the same time. So that would be the similarity. But the difference would be that because the left ventricle is stronger than the right ventricle, so the pressure generated by the left ventricle would be higher compared to the right ventricle. So your answer should include these 2 points. Number 1, peaks or contractions at the same or similar time, or you can say same or similar pattern, because both right and left ventricle contract at the same time. But the difference would be lower pressure in the right ventricle compared to the left ventricle. The next part of this question is use information from figure 3 to calculate the heart rate of this dog. So here's the figure 3. So if you look at the bottom, this data is for 1 second. And here we can see that this is the heartbeat. So if you start from here, so the heartbeat starts from here. It goes all the way up and back to the same point, which is this. So there will be one heartbeat, then another heartbeat will be up to here. So we'll have to calculate how long it took to have one complete heartbeat. As you can see from the graph, so up to here is 0.2. So obviously this will be 0.1 and that would be 0.3. And then we have 1, 2, and 3 small scales. So we need to see how much is one small scale. So we know that 5 scale up to here is 0.1 and there are 5 small scales, 1, 2, 3, 4, 5. So if we divide 0.1 by 5, so we get 0.2. So that means these 3 scales will be up to 0.6. So total up to here would be 0.36. So it will take 0.36 seconds for one heartbeat. And then we can calculate in 60 seconds there will be how many heartbeats. And that will be 1 over 0.36 times 60. And that would be roughly 167 heartbeats per minute. Okay, so next we have question number 4. So we have a bit of background of these questions. And it says anthocyanins are colored pigments found in the cell vacuole of some plant cells. Anthocyanins cannot move across undamaged cell membrane. So that means if the cell is intact, they will remain inside the cell. They will be released only when the cell is damaged or the cell membrane is damaged. Otherwise, they will stay inside the cell. A student investigated how to extract anthocyanins from blueberries. She makes 10 grams of crushed blueberries with 100 cubic centimeter of extraction solvent for 1 hour. She investigated 3 different extraction solvents, E which included ethanol, water and acid, F which included ethanol and water only, and G was only water. Okay, so the question is, when making up the extraction solvent E, the student used a volume ratio of 70:31 for ethanol, water and acid. Tick one box that shows the most appropriate volume she would use to make up 100 cubic centimeter of extraction solvent E. And here we have the 4 options. But let's do our own calculations first. Okay, so we have a ratio of 70:30 for ethanol, water, and acid. So the sum of the ratio is 101. So how much ethanol we will take? So ethanol will be with the proportion of ethanol divided by sum of ratios and times, because we want to make 100 cubic centimeters, so times 100, and that will give you 69.3 cubic centimeter. For water is 30, so 30 over sum of ratio 101 and times total, and that would give you 29.7. And finally we have acid, and the proportion of acid is 1 over sum of ratios and time 100, and that will give you approximately 0.99, which is almost equal to 1, and that answer matches second option. So our second option will be the correct answer. Okay, the next part of this question is, the student kept constant the mass of fresh blueberries, number 2, volume of extraction solvent, and number 3, the time for mixing the mixture to stand. Name 2 other variables the student should have kept constant during this investigation. So you can include any 2 points from this list. So the other variable that should be constant is the temperature, agitation, or mixing. If he was not mixing constantly, that could give you variable results. And number 3 is the source, age, or type of the blueberries. Blueberries from different sources or of different age or different types, they can give you different results. So they all should be from the same source. Crushing of the blueberries, if they are not properly crushed, then you can get variable results. And number 5, rinsing of blueberries prior to mixing, just to make sure we clean off any impurity. And number 6, consideration of ethanol or acid used. The next part of this question is, after 1 hour, the student filtered the sample. She placed the filtrate in a colorimeter and measured the light absorbance. Her results are shown in figure 4. So here we can see the bar chart. Here we have the absorbance. We can see the absorbance of E is the maximum, then F, and finally G, that was our water as a control. Now the question is, use your knowledge of membrane structure to explain the results in figure 4. So that question is for 4 marks, so you should include these 4 points. So the number 1 is higher absorbance indicates more anthocyanin, obviously because we have higher absorbance. Here in the first case, it has more concentration compared to the others. So higher absorbance indicates more anthocyanin. Or it could also mean that higher absorbance indicates more membrane damage. It mentioned in the question that anthocyanin is retained by the intact cell. So if the cell is damaged, then it's leaked out. Or you can say G0 because some anthocyanin released when blueberries are crushed. Or you can say G0 because some membrane damage when blueberries were crushed. Number 2, more membrane damage permeability results in a more anthocyanin release. Obviously, there's more damage, so that will release more anthocyanin. Number 3, E and F greater than water because phospholipids dissolve in ethanol. And because both E and F have ethanol, so the phospholipids will dissolve more in the E and F. And then E is greater than F because it has acid as well, and acid denatures membrane proteins. Okay, the next part of this question is, a different student did this investigation. He did not have a colorimeter. Describe a method this student could use to prepare color standards and use them to give data for the total anthocyanin extracted. So what the student will have to do is they have to make standard solutions of known concentrations and then compare the results with these known standards. Because this question is for 3 marks, so you should cover these 3 points. So the number 1, use known concentration of blueberry juice or extract, or you can say use known concentration of anthocyanin or pigment solution. Or you can say use known concentration of extraction solvent to be added to blueberries. Point number 2, prepare dilution series. So just like you see in the diagram. And number 3, compare results with color standards to give score or value or the concentration, whatever. Okay, so let's move on to question number 5. So this question says, describe the role of DNA polymerase in semi-conservative replication of DNA, and that is for 2 marks. So that's a pretty straightforward question. Okay, so here is the DNA. So the first step in the DNA replication is that both the strands should separate. So we can see here both the strands have separated, and then each of the original strand is used as a template. On this template, nucleotides are joined together to make the new strand. And this is the DNA polymerase, which actually joins these nucleotides onto the new strand. So your answer should include any of these 2 points. Number 1, it joins adjacent DNA nucleotides. Number 2, it catalyzes condensation reactions because joining this nucleotide is a condensation reaction. And number 3, catalyzes formation of phosphodiester bonds between adjacent nucleotides. So phosphodiester bonds are basically the bonds between the adjacent nucleotides. The next part of this question is figure 5 shows a percentage of rat cells undergoing DNA replication. Some cells contained a protein called cyclin D and some cells didn't contain cyclin D. All cells were in early interphase at time 0. So as you can see here, there's a time 0. So all the cells were in early interphase at time 0. It took less time for 25% of the cells with cyclin D to be undergoing DNA replication than for 25% of the cells without cyclin D. Use figure 5 to calculate this time difference as a percentage decrease. The solid line is the cells without cyclin D and the dotted line is cells with cyclin D. And as you can see here, using cyclin D decreases the time for undergoing replication. That means that the cell starts DNA replication and eventually cell division earlier compared to the cells without cyclin D. So now all it's asking you is how much difference it makes. So you can find the difference between these two if you start from here and go all the way up to here and you see how it is the difference. So if you look here, this is 10, this is 20. So obviously the halfway between would be 15 and halfway between 10 and 15 would be 12.5 and similarly halfway between 15 and 20 would be 17.5. And then you should also be able to know how much is this one small scale. So the difference between the two is 2.5, and because there are 5 small scales, so 2.5 divided by 5, that will give you 0.5. So that means each small scale is equal to 0.5. So using that, you can calculate the distance between this point, and that will give you 5.5 hours. So there is a difference of 5.5 hours, and the highest difference you can find is between this point and this point. So you should be able to find the distance between these 2 points. That would be about 6.1 hours, and then all you have to do is just convert them into the percentage. So this point would be at 19 hours and this point would be at 13.5 hours, and the difference is 5.5. So 5.5 divided by the original time, which is 19, and then time 100, approximately 30%. Similarly, you can calculate the other one. The next part of this question is cyclin D stimulates the phosphorylation of DNA polymerase, which activates the DNA polymerase. Describe how an enzyme can be phosphorylated. Just a simple straightforward question for 2 marks. So it's just asking you how an enzyme can be phosphorylated. An enzyme can be phosphorylated by the addition of the phosphate group. So your answer should be number 1, attachment or association of inorganic phosphate to the enzyme, and number 2, released from hydrolysis of ATP or released from ATP to ADP and inorganic phosphate. The next part of this question is some tumor cells contain higher than normal concentration of cyclin D. Use figure 5 to suggest why higher than normal concentration of cyclin D could result in a tumor. So obviously, because cyclin D increases the number of cells undergoing DNA replication, which in turn starts the cell cycle. So increasing cyclin D concentration will stimulate more cells to divide. So your answer should include any 2 points from these. Number 1, shortens interphase, or cells begin DNA replication earlier, or you can say DNA replication starts faster. Point number 2, faster cell cycle or division or multiplication or mitosis, or you can say uncontrolled cell division or mitosis. And point number 3, this results in a mass group of abnormal excessive cells.